Today we're going to look at genetic engineering and the technologies with DNA. The other day we did RFLP and gel electrophoresis, and while that is technically manipulating the DNA, it's not really engineering it or changing it. So our focus now is going to be on genetic engineering. And genetic engineering is all about not only manipulating the DNA of organisms, so that the organism is helpful. It's about actually like inserting new genes into organisms or transferring genes from one organism to another. This is going to make something called a transgenic organism, and you know transgenic organisms as GMOs. So transgenic organism means that it's an organism whose genome has been altered. And GMO, of course, stands for genetically modified organism. There are some key terms that come up with this, the first of these being a cloning vector. And what you have to understand is that a vector is a term for anything that can carry something. In this case, in biotechnology, when we're talking about a vector, we're talking about some piece of a chromosome or some chromosome that can host a new piece of DNA. It's where we insert that piece of DNA that we're trying to move to another organism so that that other organism can house it and express it and copy it into its new cells when it does mitosis. The most common cloning vector in biotechnology is the bacterial plasmid, and the bacterial plasmid is what you see here. It's just a chromosome in DNA in bacteria that's just in a ring shape. And we saw that when we looked back at bacteria back in the cells unit. A couple other terms that become important here are donor gene. All right, donor gene, that's the gene we're trying to move. Now, we're looking specifically here today at an example of insulin, right? We've talked about diabetes, and we've talked about the fact that we need a safe, clean source of insulin that diabetics can rely on. So for our purposes today, we're going to talk about our insulin gene as the donor gene. And of course, we have restriction enzymes. We talked about these the other day as well with recombinant DNA, or excuse me, with RFLP and gel electrophoresis, and we said these are enzymes that will cut the DNA at specific points. What we didn't mention is they make something called sticky ends, and I have a picture down here of what sticky ends look like. So here is a restriction enzyme sequence, the recognition sequence of TTAA, right? Now, here's the thing. The restriction enzyme wants to cut when it sees TTAA, but remember that the strands of DNA run anti-parallel. In other words, they run in opposite directions. So the cut doesn't go all the way across. It only goes through one strand. The TTAA runs in the opposite direction on the opposite strand, so you get a cut on the other strand in a different spot. It's not a blunt cut straight across. What you get are these stair-step shaped cuts that you see here. Those are called sticky ends because what you have is these open hydrogen bonding spots with the T's, and the A's, where you can actually insert something in here and it will hydrogen bond in. And these sticky ends are really, really important to biotechnology because if you can't hydrogen bond something in, then you can't insert the gene here. So this is going to be useful for inserting genes. All right, so this process that we are looking at now for genetically modifying organisms is called recombinant DNA. Recombinant DNA. And if you look at the word recombinant, you can kind of see recombine. It's a really easy process. Okay, so we're going to, instead of human growth hormone, remember we're going to do human insulin, okay, for ours. All right, so we're going to look at human insulin. But the first thing that you do is you take a human cell and you isolate what we call the donor gene, right? And here is EcoR1. EcoR1 is a common restriction enzyme. So we use EcoR1 to cut the human chromosomes from this cell and pull out the gene, and we'll call this the insulin gene. So we've got our donor gene. Next step is to take a bacterium. So here's our bacterium. Here's this little plasmid, and we pull it out, and we cut it with the same restriction enzyme. And the reason why we're doing that is we want the sticky ends of our donor gene to match the sticky ends of our cloning vector. So this is the cloning vector, and we're going to cut the cloning vector and the donor gene with the same restriction enzyme. That's going to open up our plasmid and allow us to insert this donor gene right there into the plasmid, which is what you see right here. So notice the plasmid here just gets a little bit bigger because now it's got a new gene inserted. Then you insert this back into the bacteria, and that bacteria carries out its life cycle, goes through mitosis just like any other cell would, and now through one transformation process, and that's what this whole process of recombinant DNA is called, it's transformation. Through one transformation process now, if we just culture this and let it grow up, it becomes hundreds, thousands, millions even of bacteria that are all now transgenic or genetically modified to produce human insulin. And now our bacteria will produce that insulin for us. We can purify the insulin and get that, and voila, you've got human insulin. So you have a clean, safe source of laboratory-derived insulin just from this process. So that's kind of nice. It's nice to do this because we're able to then get clean medicine for people who need it. Excuse me. The reason why we use bacteria in this process is because bacteria are cheap, they don't eat much, they don't take up much space, so it makes them ideal for this. Plus, this little plasmid is super simple and easy to manipulate. Now, there are lots of uses for this technology. The first is, of course, that of medicine and pharmaceuticals, right? Medicine, pharmaceuticals, that's what we just did. We engineered a bacteria to make medicine for us. There's also some really neat uses of this in environmental cleanup. When there's an oil spill, it turns out there are some bacteria that naturally occur in environments that are capable of eating oil, and so they are capable of cleaning up environments for us, which is really nice. We can engineer bacteria to be even more efficient at cleaning up oil spills by eating the oil that's been spilled. And it's a nice natural way of getting rid of the oil without using expensive chemicals and things like that. There are two other really important uses, and one of them is gene therapy, and this is a picture of gene therapy here. Gene therapy in humans is the ability to take somebody with a genetic disorder, usually because they've got some sort of, they were born with some sort of DNA mutation. And we can actually take their cells and create, instead of a bacteria this time, we can create a virus that is our cloning vector. And sometimes cloning vectors aren't bacterial plasmids. Sometimes they're viruses. Sometimes they're even yeasts in a laboratory. But we can take and make a cloning vector, a virus into a cloning vector, and infect these cells from the patient with a virus that's been engineered to contain the correct form of the gene that the patient has the mutation in. Then all you got to do is stick those cells back into the patient, and you can actually potentially correct genetic disorders. So if somebody's born with something like sickle cell, we one day might be able to do this and fix sickle cell, which would be a really nice thing and would improve the quality of people's lives. Right now there's only one gene therapy technology that's gotten to the testing point in humans, and that's a nasal spray that contains little viruses like this. So rather than pulling out cells from a patient, they're just inserting the virus directly into the patient's nose with a nasal spray to try to correct cystic fibrosis. The last good use of this is in agriculture, and you're going to read an article about agriculture and recombinant DNA. But I wanted to show you this too because this is very similar to what you just saw. In fact, the first steps are the same. You start with this time something called an agrobacterium, which is a bacteria that is capable of infecting plant cells. You engineer it, okay, so you're going to actually, say you want, I don't know, a drought-resistant gene put into a plant. so that the gene can survive longer without water. You engineer the plasmid in this agrobacterium to contain that drought-resistant gene. And then from there, it's very much like this gene therapy. You allow this bacterium to infect a plant cell. And that bacterium is going to put the new piece of DNA in it. Then you can grow these cells in a culture, and the plant will grow up with the new trait. So both gene therapy and recombinant DNA, or excuse me, agricultural plant transformations, that's what that's called, is transformation. Both of these things are just taking the recombinant DNA technology that we just looked at a step further. One little side note is, where did this ability to do this gene therapy come from? And this is where we do need to give kind of a little nod to the HGP, or Human Genome Project. This was a long-term international project where groups of scientists from all over the world worked together to create maps of all of the human chromosomes. And they accomplished this by sequencing genes. The reason why this is important is, if we didn't know, for example, where that cystic fibrosis gene occurred on which chromosome, we'd have no way of knowing how to fix it. Now that we know where it is, we know how to target that gene in people, and we can potentially fix a genetic disorder like cystic fibrosis. Sickle cell, again, we didn't know before the Human Genome Project where that gene was. So that's a nod to the Human Genome Project. It was meant to be, I think, a 10 or 15-year study, and it was finished early just through international cooperation. So lots of scientists worked on it. There is a picture in the classroom of the Human Genome Map that the Human Genome Project came up with. All right, last big technology, and that is cloning. Now, again, cloning is a hot button issue. I'm going to tell you how to do it. I'm not going to tell you whether or not you should do it. Specifically, you're going to be tested on reproductive cloning. Now, the first example of reproductive cloning, and that is cloning to create a whole new organism that is identical to some other organism. The first was Dolly the sheep. Okay, so Dolly. There are other types of cloning, and I will post a short little animoto about the types of cloning. Some of them are actually natural. When you think about it, mitosis is cloning. But this is an easy process. It's called the nuclear transfer process. So here we have a donor female. This is the female we want to clone. Okay, we like her genes. We want more of her. So we remove a cell from her udder, and we remove an egg from a female that we like as a mother. Okay, so we remove this egg, and what we do first is pull the nucleus out of this egg, and then you can take this udder cell and this egg cell, which now has no nucleus in it, and you can use an electric shock to fuse the two cells together. Then you can culture them in a petri dish, implant them in a third sheep, a sheep who's a surrogate mother, and she will actually give birth to a baby sheep, and this sheep is genetically identical to our original donor sheep. Okay? Super easy, however, not a very efficient process. I think a few years ago there were scientists in Korea that were doing this, and they were saying for about every 200 clones they tried to create, only 2 of them would have been viable babies, and they were using animals. I can't remember which animal they were trying to clone. But it does work. Notice this is done without stem cells. Now, there are other kinds of cloning out there that would work with stem cells. Cloning with stem cells is controversial for lots of reasons. And we've talked about those reasons in class before, getting the stem cells, things like that. But at any rate, this is how it's accomplished, and it is a really, really simple process. And hopefully tomorrow we'll get to look at a little video on stem cells and cloning. But that's biotechnology.